Welcome to part 2 of 4, looking at chapter 11. So we're looking at nucleic acid structure, DNA replication, and chromosome structure. In the first part of this chapter, we focused on kind of the history behind how we identified that DNA is that genetic material. So we identified our genetic material. This part, we're going to look at the molecular structure of our DNA. We're also going to kind of look at RNA a tiny bit because it's another type of nucleic acid that we're really going to be using in chapter 12. So in 1953, the DNA structure was solved by people named James Watson and Francis Crick. There's also another guy, Maurice Wilkins, that was involved in this discovery. So they proposed the structure of DNA as a DNA double helix. And like all good discoveries, they did not do this on their own. They used information from other scientists, so other colleagues at different universities. So we're going to go over kind of the information that they got from the other scientists. So we have Erwin Chargaff, Linus Pauling, and Rosalind Franklin. So Erwin Chargaff, he contributed to the discovery of the DNA double helix by showing that the amount of the adenine base equals the amount of thymine, and then the amount of cytosine equals the amount of guanine. And he found that this was consistent in all different types of organisms. So table 11.1 shows some of the different organisms he collected DNA from, and then he just determined the percentage of adenine, thymine, cytosine, and guanine in that DNA that he collected. So he collected DNA from organisms. We have E. coli, which is a bacteria, Streptococcus pneumoniae that we talked about in the first part of this chapter, which is also a bacteria. He collected DNA from a Saccharomyces species, which is a yeast or a fungus. He took DNA from turtle, salmon, chicken, and humans. So we have reptiles, fish, birds, and mammals up here. And then if you look, he took that DNA and determined the percentage that was adenine, thymine, guanine, and cytosine. So if you look at the very bottom and look at human DNA, it's about 30% adenine, 30% thymine, and then 20% guanine, 20% cytosine. And if you look at other organisms, so for example, let's see, the E. coli up at the top, it has about 25% adenine and thymine, 25% guanine and cytosine. So you can see that we have adenine equals the amount of thymine, cytosine equals the amount of guanine. And these are called Chargaff's rules or complementary base pairing. So we'll see these rules show up a lot in the next 2 chapters. Besides Chargaff, we have a guy by the name of Linus Pauling. He contributed more of a modeling method to how Watson and Crick could model their DNA structure. So Linus Pauling, he worked more with proteins. So he correctly proposed a model for the alpha helix of proteins. And if you remember way back in chapter 3, we talked about how proteins fold together. The alpha helix and the beta pleated sheets, those are part of the 3D folding of proteins. So how he modeled it is he used a ball and stick approach. So you have little balls that represent all the different atoms, and then the sticks in between the balls represent the bonds between those atoms. If you've taken a chemistry course, you probably have had the modeling kits, which are based on this ball and stick model. So Watson and Crick, they use the same approach to model their DNA double helix when they finally figured out kind of how things fit together, and then they actually built this really large DNA structure using the ball and stick method. And then the third person that contributed quite a bit was Rosalind Franklin, who doesn't get as much credit as she deserves, but granted, this was back in the 1950s, so you kind of have to think about the time and the society and how it was back then. So Rosalind Franklin, she worked with something called X-ray diffraction. What she did was basically she extracted DNA from living things, and we'll extract DNA in lab if we haven't done it already. It's pretty easy. She took that DNA that she extracted and she passed X-rays through it to get this 3D picture. And then based on the picture, using a lot of math, she was able to relate the pattern of her X-ray of the DNA to a 3D structure, and how that X-ray could get that certain pattern. She unfortunately passed away very, very early in life from cancer, which they think came from working with the X-rays. So it was really unfortunate for her. And she, again, doesn't get as much credit as she deserves for this. But if you look at the circle picture up here, that is the image that she created using this X-ray diffraction technique. And it doesn't really look like a lot, but you can kind of see this X repeating pattern. So the black lines make kind of an X in the image. And this is how she did her X-ray diffraction technique. She took those DNA fibers that she extracted from living organisms, passed X-ray through it, and then she got this really neat repeating pattern from X-raying the DNA. So what happened is that Watson and Crick, they took information from these 3 different scientists. There's a lot of stories about kind of what happened behind this. A lot of people say that Watson and Crick, they stole data from Rosalind Franklin, or she shared it and she didn't really know what she had. But anyways, Watson and Crick, they built out this DNA model based on that X-ray diffraction picture. They used that ball and stick method. They knew that adenines match up with thymines, cytosines match up with guanines. So they came up with this idea of the DNA double helix, and they awarded the Nobel Prize in 1962 for their discovery. Unfortunately, Rosalind Franklin, she passed away right before they awarded the Nobel Prize, and they can't award it to people that have died. So that's why she's not really as much of a household name for this discovery. And this link down here on the bottom of the slide, I'll post the video on the e-courses so you can watch it. It kind of gives you more of the backstory of how this DNA was discovered, and it has interviews with Watson and Crick and kind of what they thought at the time for that. So this DNA double helix, as we learned way back in Chapter 3 again, you might want to go back and kind of review Chapter 3 looking at the nucleic acids. But nucleic acids, they're made up of monomers called nucleotides. So we are going to look at the nucleotides. So these nucleotides, they're going to bond together using the phosphodiester bonds that we learned about again in Chapter 3. And those nucleotides, they form single strands. And we have to have 2 of these single strands in order to form our double helix. That's on the bottom of this image. And this double helix, this is the part that Watson and Crick figured out. That double helix part of it, at the end of this chapter, we're going to look and see how this DNA associates itself with proteins to form chromosomes. So remember, part 1, we talked about chromosomes are made up of proteins and DNA. And we find DNA in the form of chromosomes within cells. But we're going to kind of start back at the nucleotide part and then work up to these chromosomes. So nucleotides, those monomers or those building blocks, they have 3 components to them. So they have the phosphate group, a pentose sugar, 5-carbon sugar, and then 1 of 4 nitrogen bases. So DNA has a phosphate. So if you look on the left-hand side, we have 1 nucleotide here. So we have the yellow phosphate part. For our pentose, or 5-carbon sugar, we have a sugar called deoxyribose. So it's a really simple monosaccharide or simple sugar. And then to that sugar, you can have one of 4 bases. You're going to have adenine, thymine, guanine, or cytosine. If the base has a double carbon ring, it's going to be a purine. Those are the adenines and guanines. If it has a single ring, single carbon ring, it's going to be a pyrimidine. So that's the thymine or the cytosine over here. So DNA, it has a deoxyribose sugar. It has adenine, thymine, guanine, and cytosine. And I mentioned I'm going to kind of throw RNA in here a little bit just to kind of look at the similarities and differences. So RNA is also a type of nucleic acid. It has nucleotides. They look very similar to the DNA nucleotide we just looked at, but instead of deoxyribose, they have a ribose sugar. So it's still a 5-carbon sugar, it's just a ribose. And then for the bases, you're going to have adenine. Instead of thymine, you're going to have uracil. So you have the uracil one. Then you're going to have guanine and cytosine. So similarities and differences between DNA and RNA. DNA has deoxyribose sugar. It has thymine as one of its 4 bases. RNA has ribose sugar, and it has uracil as one of its 4 bases. So on these nucleotides, in chemistry, there's a conventional numbering system that you can use to identify different carbons or different molecules within a molecule. So if we take a DNA nucleotide, shown up here, we know it's DNA because it has a deoxyribose sugar and it has a thymine base, so we can actually identify it. If you look on the deoxyribose sugar, the carbons are numbered 1, 2, 3, 4, and 5. And it's actually 1', 2', 3', 4', and 5' carbons. So this numbering system is going to be important later on when we look at how our 2 strands of DNA fit together to make the double helix. But the 5' carbon is always attached to the phosphate. So 5' attached to the phosphate group. So that's a really good way to kind of remember this numbering system on our pentose sugar. Okay, so we're going to take those nucleotides, we're going to hook them together using a phosphodiester bond, and we're going to form a single strand. So this is a DNA single strand. So if you look at the top of this DNA strand, we have a thymine, so you see the phosphate, the blue sugar, and the green thymine. That sugar is attached to the phosphate on the next nucleotide by that phosphodiester bond. So now we have an adenine nucleotide as our second one down. Our third one, cytosine. Our 4th nucleotide, we have a guanine that's attached. So make sure you can see each of those 4 nucleotides. You'll see the phosphate, the sugar, and the base. And then phosphate, sugar, base, and so on. So this is where that numbering system shows up again. If your phosphate is sticking out, that's called the 5' end of your strand. So up at the top, that's our 5' end because our phosphate is sticking out. The other end of the strand is called the 3' end because the 3' carbon is facing out on that end. So the 5' and the 3', make sure you know how to identify those and kind of what they mean, because we'll see them quite a bit in the next 2 chapters. We then take 2 of those single-stranded DNAs, we hook them together by matching up the different nitrogen bases with each other using Chargaff's rules. So DNA is double-stranded. We have 2 of our single-stranded molecules. It forms a helix, so it has a helical shape to it. The sugars and phosphates form a backbone. So they're now represented just as the red strands in the picture up here. The nitrogen bases, they face in towards each other, and those nitrogen bases are held together by hydrogen bonds. And if you remember in chapter 2, hydrogen bonds, they're really weak bonds that form between molecules, but if you have a lot of them, they're very, very strong. So DNA has a lot of hydrogen bonds, which keeps the DNA together But those hydrogen bonds, they can break pretty easily, so we can still separate our two single strands from each other. And that'll be important when we get into replication and then chapter 12 as well. And then we have the base pairing with Chargaff's rules, so the specific pairings we're going to see when we have this double helix. So if you remember, adenine pairs with thymine and guanine pairs with cytosine. So those are the Chargaff's rules or the complementary base pairing rules. So if you look at the image, we have on the left-hand side, we have a thymine nucleotide. On the right-hand side, it's paired up. So we have hydrogen bonds to an adenine. So A's and T's go together, C's and G's go together. For each of the turns in our double helix, it's 10 base pairs. The two DNA strands that are hooked together are complementary to each other. So again, Chargaff's rules. So if we have 5 prime, we G, C, G, G, A, T, 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 3 prime. It'll go 3 prime, C, G, C, C, T, A, 5 prime. So we again see the G's and the C's go opposite of each other, the A's and the T's go across from each other. The other thing is that the 5 prime and the 3 prime, they're anti-parallel to each other on the two strands. So one strand runs 5 to 3, the other strand goes 3 to 5. So they're facing opposite directions of each other. So just some really important information about our DNA double helix. So we saw our DNA double helix kind of represented as just like a flat model. You can also represent it using a space-filling model that will show what are called major and minor grooves. So you have big areas and little areas, or minor grooves. And you can see that on the image on the right-hand side. The major grooves, this is where proteins can fit into the DNA. They'll bind in there. They'll help wrap that DNA up into chromosomes. They also can turn genes off so that they're not expressed, or it can turn genes on so they are expressed. So there's certain parts of the DNA that help with the expression of those traits that are coded for in our DNA molecule.